Today, I'm really delighted to introduce our speaker tonight, Dr. Rebecca Lyons. I first met Rebecca last year, actually. Rebecca teaches a month-long class here at SNARL every year. She's from University of the Redlands, and they have a course called Environmental Chemistry. And her students, you guys can wave your hands. They're all right here in the front. Sporting their university wear. I really like that. It's very fashionable. And I would invite everyone in the audience to ask them questions after the seminar, because these students are spending the entire month here learning about environmental chemistry with Rebecca. They're great students. They're really knowledgeable. And I would encourage you to pick their brains a little bit. And so Rebecca tonight is going to be talking about a subject that is, I think, relevant to us all here, endocrine disruptors in the wind and how these persistent organic pollutants travel across California. So, Rebecca, I'll let you take it away.
Okay, thank you. So exciting. Wow, what a turnout. I'll tell you, for a chemistry lecture, this is impressive. Usually drives people away. So thank you very much, Carol, for inviting me to do this. I'm really excited to share my work. We've had some really amazing presentations. And they're all from people that you know where they came from, Forest Service folks, national parks, UC people. But I'm from the University of Redlands, and so before I get into the work that I'm doing up here in the Sierras, if you could call it work, let me tell you a little bit about the university and what we're doing up here at SNARL, and then I'll get into the nitty-gritty, which is a little bit about endocrine disruption, what the endocrine system does for us, what it is, the compound that I'm looking at specifically, a little bit about the area. You guys are more knowledgeable about this than I am probably, so I'll give you the short tour of California, and where nonylphenol distributes in this area. Some weird stuff going on with the snowpack, climate change, and sort of where we stand in the state of regulatory affairs. That's the fun stuff. Okay, so here we go. Who's the University of Redlands? We're about an hour outside of Los Angeles, just up against the San Bernardino Mountains. It's a small, private, liberal arts university. We have about 2,400 undergraduates. There are 10 of them here. And right, we represent. I'm actually in the chemistry department. A lot of people think I'm environmental studies, but I'm actually trained as a chemist, a physical chemist, actually, and then environmental chemistry is sort of my specialty. So what I think Redlands does really good in the chemistry department, we have 9 professors. For a student to graduate with a chemistry degree, they have to do an independent research project for at least a year. Usually it's a year and a half to 2 years, one-on-one with a professor as an advisor. So it's a lot like being in graduate school. They write very thorough thesi. They do a capstone presentation, and a lot of them go on to publish their work in major journals. So we do a really good job with that. The other thing that I think we do really well is GIS. We're in the same town as Esri. I'm sure a lot of you have heard of them. And we work pretty closely with them. We have pretty close affiliations with Esri. We also have a Center for Spatial Studies. I happen to be the chair of the Spatial Studies program right now, so this is another hat that I wear, so I had to give a little plug for that. And we have a master's program in GIS as well. So in terms of resources, a lot of people are like, well, you're not at a big UC school, you don't have a lot of resources. Au contraire, I have many resources, and 10 of them actually are sitting in the front row tonight. So, okay. So the class that these guys are in is actually called Mile High Chemistry, and there's Sebastian checking out some of the weird chemistry in Mona Lake. And so what we're doing is each of the groups gets to study a particular watershed for a lake or a body of water. So I think somebody's doing Convict Lake this year, somebody's doing Parker Lake this year. And what they do is they accumulate all the data on that lake. They do atmosphere, soils, water, the whole enchilada, put that together in a big report. And we've shared that historically with Inyo National Forest. But we're talking about putting together a more solid database that is GIS accessible for folks doing research here at SNARL. So that's sort of where we're heading with all of this. We've got 5 years worth of data now. I've been doing this class for 5 years. And so to be able to put that all together and see like long-term trends, climate change, all of that, and the data that we're accumulating should be pretty interesting. So I'm excited about that. Okay, so a little bit about endocrine disruptor research and how I got here. So I actually graduated from the State University of New York in 2010, December, and moved out here to Southern California. And, you know, as a new professor, they say, okay, well, we want you to put together a research program. And you're, deer in the headlights. What am I going to study? So in Southern California, I thought I'll go for the low-hanging fruit, and I'll look for pollutants in the water in Southern California. That should be an easy one. And I actually specialize in trace organic pollutants. So I was like, okay, I'll start poking around. And I had a student, and she said, well, I want to study something in the water too. I said, well, okay, why don't you look for this endocrine disruptor? Never thinking that I was going to find it. And behold, there it was. Well, then I was having a hard time finding a lake that didn't have this endocrine disruptor in it. I was like, holy smokes, this stuff is all over the place. I have a good friend at UCLA, Dave Caron, and he said, well, go to the Sierras. You'll find clean lakes up there. So here I am. This is how I found the Sierras. I kind of stumbled into it and started poking around lakes up here looking for these endocrine disruptors that I study. So that's the backstory. So before I get into this specific endocrine disruptor that I'm looking at, I just want to give everybody a little bit of an overview of what the endocrine system does for us. It's actually a collection of glands. I know there's probably some biologists in the room that could give a much better description, so I'm a chemist, keep that in mind. I'm just going to give the overview. Collection of glands in your body that is an information highway, basically. So that sends signals back and forth through your body, telling it when it's hungry, telling it when it's full, regulating your metabolism, growth, reproduction, everything. It regulates all of those things. The interesting thing about the endocrine system is all of those systems that are in place are feedback. So if one of them is going up, the other one goes, oh, okay, I can sort of ramp down a little bit. Or they're talking to each other, basically. Another interesting thing about it is that it's communicating with chemicals that's excreted into the bloodstream on the order of parts per billion. It's an extremely sensitive system. So it just takes a little tweak, a little imbalance here or there for the endocrine system to be all out of whack. It's kind of like a big mobile that you're hanging over a kid's crib, and you touch one and everything else has to adjust to make up for that. So it's a very sensitive system. Okay, so what's an endocrine disruptor? How do we mess up a perfectly good system in the human body? Well, the way hormones work, and I'm going to switch over to my laser pointer here. I've been informed that this is actually a cat toy. Here we go. Okay, so if you have a cell, this is the cell wall right here. On the surface of the cell wall, there are receptor sites. And when you excrete a normal hormone, it fits like a little lock and key into that receptor site, and it initiates a cellular response. So the cell might turn on a segment of DNA or start synthesizing a certain protein. So there's going to be some sort of response in the cell. If you have an endocrine disruptor, it also fits into that cellular receptor site. And it can do a number of different things, actually. Sometimes it will initiate the response that the hormone would normally do. So it would just start down that pathway. And you might accelerate or exacerbate a certain pathway. Another thing that it might do is it might just gum up the works, like throwing sugar in the gas tank, and it stops the entire sequence of events. It just stops. And then another thing that happens is you get leftover hormones that should have fit into that receptor site that are now floating around in your bloodstream and sending feedback, like I was talking about, to sort of shut that system down. So there's a bunch of different things that can happen when this happens, when you get a disruptor. So it's going to significantly alter all sorts of things: development, metabolism, physiology, all sorts of stuff can go wrong. Okay, this is a really busy slide, but what I wanted to do is actually just point out a couple of examples of endocrine disruptors that you may have heard of, that have made the news, and show you a couple of the different categories of endocrine disruptors that are out there. So there's DES, which was actually a medication prescribed to women who were pregnant to keep them from miscarrying their children. It actually was an estrogen mimic, so it acted like estrogen, and it would cause developmental abnormalities in especially the daughters. They're called DES daughters, and a lot of them were infertile themselves when they came to childbearing age. So that's one of them that people have heard about. People also know that there's naturally occurring endocrine disruptors out there. There's genistein, which occurs in soybeans. So a lot of people who are worried a little bit about maybe getting too much estrogen have been warned off of soy and soy products. Well, this actually acts like estrogen and also blocks the behavior of testosterone. So it has sort of a dual action. So people have heard about that one. Recently, I think this was about maybe 7 or 8 years ago. People probably heard about BPA, bisphenol A, in the news. It's a plasticizer. It gives things sort of a nice soft texture to the plastic. They were putting them in things like baby bottles, the blood bags in blood banks, that sort of thing. And it also mimics estrogen, and people were finding fairly strong responses to this BPA. So now when you buy a water bottle, look at the bottom of it. It'll say on the bottom, BPA free. A lot of things will say that on it now, so people caught on to that. There's a lot of pesticides and fungicides that are also endocrine disruptors, and this is one, I pointed this one out in particular, this polychlorinated biphenyls. Those were actually banned in the 1970s, but those are what we call a legacy pollutant. So a legacy pollutant is something that we've stopped making, we realized it was bad, but it's persistent. In other words, there's no breakdown pathways for this stuff, so they're still around. Once you generate a PCB, it's pretty much in the environment ad infinitum. There's no place for it to go, really. So that's a legacy pollutant, and it's also a thyroid inhibitor. So people who have Hashimoto's disease maybe sometimes have been exposed to PCBs, for example. So those are some of the baddies. Okay, so here's what I'm studying. My endocrine disruptor is this structure on the right. This is why I'm impressed that you guys are here. I'm going to start throwing stuff like this up on the board. So here's the structure of the endocrine disruptor that I'm studying. Its name is 4-nonylphenol. And right here on the left is 17-beta estradiol, which is the most common form of estrogen. And you can see, without even being a chemist, that there's a lot of shapes that these have in common. So they're not identical, but it's close. And the interesting thing about the estrogen receptor site is that it's not terribly selective. Actually, a lot of things can bind into that site and block that receptor site. So this actually does a very good job of that. And this isomer, or configuration as we call it, is actually the most common one. So when this gets formed in nature, which it actually does, this is the one that shows up the most, and it's the one that looks like estrogen. So that's what I'm looking at. Whoops. Okay, that worked. Cool. All right, moving on. So 4-nonylphenol is an estrogen mimic, like I already said, and I want to tell you a little bit about the source of it. It's kind of interesting. It actually comes into the world as nonylphenol polyethoxylate. So it's these long chains of plasticizers, basically. It's actually a surfactant. And what a surfactant does is you can put it in another formula for something, and it makes that formula go on smooth. So if you're putting it in a lotion, you want it to go on smoothly on the surface of something. If you're doing a paint or a plastic and you want to make a thin film for food wrap, say, you put this stuff in it makes a nice smooth film. The other thing they use this a lot in is pesticides. Pesticides, you want it to cover the leaf, right? And you want it to be a nice smooth cover on that leaf. So they put it in pesticides. It's about 10% by volume of all pesticides. Not all pesticides, most pesticides. About 10% by volume. That's a lot. Especially when you start thinking about how much pesticides we use in this country. So 10%. Now, they don't have to claim that on the label, which is interesting. This is an inert ingredient. It's not the active ingredient, the pesticide, so they don't have to tell you what it is. So it'll just say inert ingredients, and a lot of the times it's this nonylphenol polyethoxylate. So the two primary means of this stuff getting into the environment are through wastewater treatment plants, because it's in a lot of personal care products, and also in pesticides. So those are the two ways that it's getting into in the environmental systems. Once it's in the environment, it goes through a biodegradation, meaning the bacteria break it down, and it goes from the nonylphenol polyethoxylate, the surfactant, to 4-nonylphenol, which is the endocrine disrupting compound that I've been talking about. It's also relatively persistent, meaning, again, it's a little bit like the PCBs where I was talking about legacy pollutants, where there's not a lot of ways for this to break down. There's not a lot of bacteria that can metabolize it or use it as a carbon source. So it pretty much stays in the environment the way it is. And then the other interesting thing about it is it does this thing called bioaccumulation. So bioaccumulation can actually happen two different ways. This stuff is very oily in its nature. So it actually wants to bind to fatty tissues, so skin, organs, that kind of thing. So we call that lipophilic. So something that's lipophilic can actually get into an organism just by like being in the environment with it. So if you're an algae and this stuff is in the water, it's going to sort of go across the cell membranes and get into the organism. So that's one way it can get into an organism. The other way it can bioaccumulate is the classic food chain where the little fish gets eaten by the big fish, gets eaten by the eagle, you know, the whole thing's going to go up the food chain that way. So just to show you a little bit of what that looks like, this is kind of a really interesting chart on the effects on wildlife. So over here we have the algae, these little guys maybe. And so some of the nonylphenol can just cross their cell membrane and get into an algae. And we see growth effects at fairly high concentrations. So if you look at that scale over there, that's actually a log scale. So pay attention to that when we start getting into more bioactive concentrations. Okay, so once we get into invertebrates, cute little Daphne there, it can start affecting reproduction at as low as 10 micrograms per liter. Okay, so it doesn't take a lot to start causing reproduction. We start seeing problems with reproduction around here, and then we get into survival issues. For the first time, you see that at a little higher concentrations. And then once you get into vertebrates, now we really see problems. It's affecting survival at pretty low concentrations now. And the lowest concentration right here is where it's affecting reproduction. You're seeing reproductive abnormalities. And so I put a little red dotted line right there where we start to see bioactivity, and that's at about 3 micrograms per liter. So it doesn't take a lot of this stuff. 3 micrograms per liter is 3 parts per billion. So now we're down into that range that I was talking about with the reproduction, the endocrine system. There are some human effects. People, of course, are very concerned about how this might affect humans. And so what people have related this to are things like impaired immune system functions, hormone-related cancers like ovarian and breast cancer, reproductive health. This is an interesting one, an obesogen. Obesogens are things that disrupt lipid metabolism, so people aren't metabolizing lipids the way they should be, and they're relating that to heavier body weight. But the thing about humans is that we're pretty complicated, and it's really hard to tell a human to just do this one thing, because they're never going to do just that one thing. They're going to do all sorts of things. And so there's lots of environmental factors that affect humans. So in every one of these studies, they're saying it has been linked to. We're not saying it's 100% causative, but it's been linked to, just to point that out. Okay, so how are these things getting around? We said that there's a couple ways that they can get into the environment. Now, once they're in the environment, how are they going to move around? Well, there's actually 2 major ways that any of these endocrine disruptors can get around the world. They can go as an aerosol themselves. So a picture of a crop duster was a great example of something being aerosolized and released into the environment. Or once it lands on the ground, it can attach itself to dust, and then the dust dries out and blows around. So now we've got this dust that's sort of moving globally in some cases. We've got a lot of dust movement these days. So in terms of how long it takes for it to move from the source to other places in the world, where it depends really. If we're going to go from the continental US maybe over to Japan, it takes about 2 weeks. It takes a little longer for it to go north to south. For it to cross the equator, it's actually very difficult. So that takes about a year for it to get all the way down to South America. But then for it to get to the top of the troposphere, the part of the atmosphere where we live, it only takes about a week. So once it gets into the environment as a dust or an aerosol, it's pretty mobile. It can start moving around pretty easily. So there's layers of the atmosphere, and the one that we live in is the troposphere. It's up to about 10 kilometers in height, and then there's a boundary, and then you get into the stratosphere. So just a different kind of layering system once you get past there. So we are in a very well-mixed part of the atmosphere, and it's the troposphere. Yep, you bet. Okay, so a little bit about California, in case you were wondering. You are here. So this is a really amazing state to study this sort of thing in. And part of the physical chemistry that I looked at before I came to California was physical barriers to movement in the environment. And we happen to have one of the coolest physical barriers to environmental movement, like right there. And that would be the Sierra Nevada range. Conveniently bisecting the state, and on the other side of the Sierra range, we have the Central Valley, where you're probably aware that they use a lot of pesticides. If you think about the major wind direction here, it's actually bringing stuff from the Central Valley towards us. And I actually have a really cool video to show you, and hopefully this is still working. So I'm just going to take a little break from this. Hold that thought. And here it is. This is real time, folks. I love this. I could stare at this all day. So this is the wind right now. The fatter the lines, those white lines, the faster the wind is moving. And it happens to be a great windy day out there, so it's a really dramatic picture. You can look at this on some days, and it's kind of like, meh, whatever. But so we're over here, and I think what's really interesting about this video is you can see the Sierra range. You guys all see it? Yeah, right there. Yeah, so some of the wind is making it over. A lot of it is swooping around and sort of coming out the other side. A little of it's going around the top. And you can actually see the Continental Divide really well, too. That's kind of neat, huh? Yeah, so when you start looking at wind patterns and associating it with physical barriers that the wind needs to get around, it's pretty cool. If anybody's interested, there's the web address. I left it up there so you guys can stare at this at home. It's kind of like a real-time lava lamp. I love this thing. You're right? Bad day to fly into Denver. Yeah. So I just wanted to show you guys that. You can really see those barriers. And I think I can, so you guys want to see more? Hang on. No, that's about as zoomed out as I can get. My resolution's not great on this. All right. Back to our regularly scheduled program. Okay, so from current slide. All right, so I mentioned that the University of Redlands has a crackerjack Center for Spatial Studies. I can walk into the office and say, hey guys, I want you to take all of the topographical high points on the Sierra Nevada mountain range and line them up and put it on a map. And they do. And that's that dark line right there. That's the ridge line of the Sierra Nevadas. And then I can say, okay, I want you to take all the pesticide data by county and color code it from the highest use to the lowest use. And so they do that for me. They're great. So again, sort of a you are here. On the other side of the mountain range, we have the darkest red. Here's Fresno County with the highest pesticide use in the state. And then we've got Mono and Inyo counties on the other side with the absolute lowest use in the state. And here they are juxtaposed in the middle of the state with this phenomenal physical boundary right in between them. And I thought, man, this is gold. Let's see what, not really, but this is great if you want to look at the movement of pesticides in the environment, in this case, the inert ingredient. Okay, so we've got that in our head.
Where is the stuff from west of Ridgecrest?
Over here, you mean, like this part of the map?
Yeah. What's being hit with pesticides?
Just the whole county.
Yeah, it's that county. Is that what you're asking about?
They're just averaging by county.
Yeah, it's an average by county. Thank you. Yeah. Yeah, sorry. Neglected to say that. Okay, so here we go. Pesticide use by county. All right, so with that map in my mind, we started thinking about sampling sites. Like, where do we want to look and see where the distribution of this endocrine disruptor is. So again, it's basically the same map without the counties. There's the ridge line of the Sierra Nevadas. And so we wanted to span either side of the Sierra Crest when we started picking sampling sites so that we can see what it was going to do from one side to the other. And when we did that, for each one of those points on the map, we looked at surface water, we looked at atmospheric deposition, and we looked at snowpack. So those were the three things that I was really focusing on. And we wanted to look and see what the different amounts of wind would do to these different sites too, because that's obviously a major player. So there's some of my students out there with a snow tube taking snow cores. It's not a bad gig. All right, so here are some of the results. Just to give you an idea of what you guys are looking at here, I want to show you that same map, okay? And I'm talking about this little cluster of sites. So this is on the east side, and it's right out the window. This is Convict Creek Canyon right here. And I started taking samples right there at the top on the far end of Lake Dorothy and worked my way down the canyon all the way to Convict Lake. Okay, so it's that little bit right there. Okay, and I'm going to back the slide up just a second so you can see the whole effect here. So here's the ridge line right there. And this is right around Lake Dorothy, about 5 miles from the ridge. And then working my way down the canyon. So this is surface water that I'm looking at. And here I am in Convict Creek. I just also wanted to point out, there's the bioactive concentration right there. So that was weird to me when I saw that. First of all, that it was even here. And second of all, because it has such an interesting distribution. Like, what's going on with that? So we started taking this apart a little bit, and I started looking at all the separate parts of what could have been coming over the mountain. And I looked at wet deposition, which is the yellow bar right there. I compared that to the amount of this compound, nonylphenol, and those are the blue lines. So you can see there's nothing up here, and then as you get further away, there's more and more of the compound that I'm looking at. And I looked at this dry deposition as I went across. And dry deposition is just dust, really, is what I'm referring to, is dust that flies over the mountain range. And the wet deposition didn't really change. So I was getting the same amount of precipitation anywhere I took a sample in Convict Creek Canyon. But the thing that was changing in each of these things was the amount of dust that was coming over. And you'll notice it's really inconsistent. So we all know that dust can be internally generated. You can walk down the street and there can be a big puff of wind and the dust swirls around and lands. So you know there can be dry deposition that's internally generated, but it was also the only thing that was different from one site to the other. So I was associating this in my mind with the nonylphenol being attached to dust particles that was coming over the mountains. So that made the most sense to me. So there's a couple of conclusions that you can sort of draw from that kind of data. First of all, that the summer influx of precipitation is consistent spatially, like I was seeing, that the dust deposition is what's fluctuating, and that more of this stuff is depositing, more of the compound is depositing further away from the Sierra Crest as you're moving east. Okay? And so we all recognize that silhouette. And there's such a dramatic uptilt to the Sierra Nevadas. So you've got this face right there that is creating almost a shielding effect on the other side of it. So if you were to go around to the west side of that face, that's what it looks like. That's Mount Whitney from the other side. And so it's a very gradual tilt on one side and a steep drop off on the other side. So my theory is that it was not depositing there. There was some sort of a shielding from the uptilt of the Sierra Crest. So I set out to look at that. I said, all right, let's see if that actually fits the data. So this was actually done by one of my students, Austin. And so I said, okay, so here's my sampling site. Here's the apex of the mountain range that I'm looking at. So I'm looking at a certain distance away from that apex, and I'm looking at, okay, how high is that head wall, and what's the slope on that head wall? Actually, this little slope should be right over here. So that should be the slope. And I said, can I put those things together mathematically and use that as sort of a predictor of where I'm going to find this compound? Where I'm going to find the compound. And I called that topographical shielding, or T. And this is actually something that Roland Knapp observed to some degree in some of his work in a 2007 paper. And I was like, well, I'm seeing that here too. So let's see if I can actually make this a prediction. And so what I did is I took the data for concentration, and I took this T value, and I applied it to 3 different canyons. And it looks like there's a pretty good connection. So I tried this in Little Lakes Valley, Convict Creek, and Morgan Pass. And I'm not seeing a lot of outliers, a little bit in Morgan Pass, but if you know the topography of Morgan Pass, it's very different from the other ones. So it's a little bit different, but it still follows that same trend. So this topographical sheltering value is actually a pretty good predictor of where we're going to find this compound. Okay, here's the surprise though. This surprised me even more, I should say. This is the snow concentrations. So this is 4 nonylphenol concentrations in snow. I didn't do this the same year as the other one, but I have surface water concentrations from the same year, and they looked about the same as the first graph I showed you. What's different about this is this axis right here. I was showing you this, and it was about 2 orders of magnitude lower in water than it was in the snow. And I've showed this to people that study snow and look at, snow, how snow transports things. And they weren't surprised by this at all. But when I saw it, I was like, holy smokes, that's a lot of nonylphenol. So we've changed it by 2 orders of magnitude now when we started looking at the snow. And all I could ask myself is, what is going on with the snow?
Is that a liter water equivalent or volume?
It's snow water equivalent. Yeah, with the snow. With the surface water, it was just straight up water. So, yeah, good question. Thank you. Okay, so what's up with the snow? I learned a lot about snow in this project. I learned that snow actually has 2 parts to it. There's a dust particulate frequently, and then there's also the snow water, so the ice crystal. And I also found out that snow is actually technically a mineral because it's a pure compound with a crystal structure. It makes it a mineral. Okay, cool. Snow does this other thing as it's falling through the air. It picks up aerosol particles. So as it's falling down, we call that rhyming. And it's picking up aerosol particles as it falls through the air. And what it also is picking up as it falls through the air are organic pollutants a lot of the time. And that's a scavenging effect that's been documented in a lot of work out there. So the snowpack effectively has become a reservoir, not just for nutrients, like a lot of people look at, but also for organic pollutants, which I didn't know about until this project came up. And so what that means is that the snowpack is accumulating this stuff over the winter, and then in the springtime, when we get spring melt, there's high concentrations, I'll venture to say high concentrations, of these endocrine disrupting compounds in the spring snowmelt. So I wanted to deconstruct the snow and see, was it coming over in the wet part of the snow, or was it just coming over in the dust? Because one of the things I thought is, well, okay, maybe it's just the dust that's landing on the snow. Maybe it's the snow itself. And so I separated those 2 things out and did a comparison on both sides of the Sierra Nevada. So here's the western slope of the Sierra Nevada, and here's the eastern slope of the Sierra Nevada. And that center line, again, is that ridge line that I've been showing you on the maps. And the way it turns out is that on the west side of the Sierra Nevadas, you're seeing a lot more of it in the snow water. So you're seeing it in the wet part of the deposition and a little bit of it in the dust. When you get to the east side, it's almost, there's a lot more of it respectively in the dust compared to the snow water. So there's less in the water and more in the dust, is what I'm saying, as a ratio.
How were you able to separate?
Those 2 things?
Yeah.
So there's 2 kinds of ways of collecting atmospheric deposition. There's a really cool device that when it senses wet deposition, there's a little door that moves over and it collects the wet deposition on one side, and when it's not raining, it goes over to the other side and lets things settle into the dry. So it's just like this, it's almost like 2 buckets with a door that moves back and forth depending on whether it's raining or snowing or not. Yeah, it's kind of cool. There's a simpler device that my students are using, and you can actually collect it all at once and filter it. So that's another way to do it. Not quite as snazzy.
But the filter lets the dust go through?
No, the filter would catch it. Lets the water go through. Yeah.
Okay.
Yep. So there's that option too. Okay, so in the Western Sierras, it's been fairly well documented that when you have your organic contaminant and it's in the warmth, it's a semi-volatile, so it'll volatilize, it'll get up into cloud systems, the clouds go up the side of the mountain where it gets colder, and they deposit out. That's kind of explaining the west side of the mountains fairly well, but it didn't really do a very good job of explaining the east side of the mountains. So I started looking at the different parts of the snow that could be transporting it and trying to look individually at all these different things that snow could do. So I thought, okay, maybe it's just the dust in the snow. And so what I'll do is I'll filter it, like I was talking about, and look at just the amount of dust relative to the amount of snow and say, okay, is it just coming over on the dust? Well, I did that, and it looked like somebody just aimed a shotgun at the wall, and it was just a complete scatter plot, had no relationship at all. So it wasn't the amount of dust necessarily, because again, there's that sort of internal generation that can happen. I thought, okay, if it's the size of the snowflake, that might have something to do with it. Smaller snowflakes are going to have more surface area for it to attach to. And so I had some poor student sieve snow so that we would have consistent sized particles of snow, and then we would expose that to aerosolized nonylphenol to see what it would stick to. This poor kid spent an entire summer in the cold room, like in his ski parka and gloves in Southern California. There's a project for you. Okay, so was it the size of the snowflake? There's some of the data from that project. And it was true up to a point. So we would see more nonylphenol sticking to it up to a point where sort of the ratio of surface area to volume would make it so that it wasn't really changing that much. So it sort of leveled off. So up to a point, that was a factor. Was it this topographical shielding? Well, we already showed that had some effect as well. And then the other thing to look at was the wind speed. Was it just the wind that was transporting it? And there's this thing called deposition velocity, which is related to wind speed. Was it deposition velocity that was making it do that? And we tested that as well with different speeds of wind to see, and this is testing in the laboratory, to see where it would settle out. And there was an inverse relationship between how fast the wind was going. And so we suspected that it had something to do with the wind as well. So how do you take all of these different pieces of data, all these different parts of how snow behaves, and sort of come up with a predictive model that will say, okay, this is where we expect to find this nonylphenol. We know it's all topographical shielding. We know it's wind. We know it's all these different things. How do we put that all together in one model? Again, I go to the GIS department and I say, okay, I want to overlay all of these different things that could be affecting where I see this compound. I want to look at topographical shielding, snowflake size, and deposition velocity, and consider the relationship that's going on between each of them. I said one of them was inverse. With topographical shielding, it was clearly an exponential slope. Is it a linear correlation? So we're going to explain the type of mathematical relationship that they have, and then we're going to put it all together in one model, and then we're going to validate it with field data. That's the plan. So, little drum roll. Ta-da! There it is. They did it. Okay, again, there's the ridgeline. And what this shows us is that there's low risk right up against the ridgeline. In certain places, there's a lot more shielding than others, or maybe faster wind speeds. There's little pockets where things either open up into more of a plain, or sometimes you'll even get sort of a capture. If you've ever looked at the way McGee Creek sort of curves around and you almost get like a wall that's facing the incoming wind direction, you might also see sort of almost a trapping effect in a place like that. So you'll see these little hot spots where it's landing in the eastern Sierras, and then as you go further and further away from the mountain range, it's kind of landing in the plains there, the lower places. And then I did a little bit of field validation by looking at some of the hot spots here versus the places where there was low risk, which was some of the data that I showed you earlier, and it correlated like 1-to-1 correlation. I should have put that slide up, but I forgot to. But yes, there's a 1-to-1 correlation there, just about like 0.95. So that was pretty interesting that we were able to say, okay, this is really working. So I just wanted to link this in a little bit with climate change, so how climate change might affect the distribution of some of these compounds globally. We have seen evidence that there's an increasing amount of transport of compounds on dust. That's happening more and more. We're seeing that. Wherever you have desertification, where you have maybe agricultural land that's been overused and is drying out, and the soil is moving, is being blown off of there, you're going to see a lot of this dust movement that's going globally. So actually, this is a picture of a dust storm off the coast of China. This is not a cloud. That's actually dust right there. Here's clouds. You can see clouds are nice and white and fluffy, and this guy's sort of gray or tan. We saw a really intense dust storm in 2015. I don't know if you guys remember some of the sunsets we were getting in 2015, partially because of all the dust particles in the air, so that's a great example of one. And what they're finding on these dust compounds is everything from organic pollutants, like the ones that I'm talking about, to heavy metals. I was just talking with Carol about mercury on dust particulates, asbestos, and even bacteria that can live on these dust particles and move with it. So we're seeing increasing evidence for all types of movement across physical barriers, including things like the ocean. So I wanted to link up some of the data that I had with different climate change phenomena that we're seeing here in the Sierras. And so I really started this work in 2012, which was, of course, a pretty big drought year here. And so what I wanted to do is say, okay, does this have anything to do with the climate that we're seeing here from year to year, or the amount of snow or precipitation that we're getting? And so in the winter of 2012 and 13, this snow depth was a little higher than it had been. So here's our maximum snow depth for 2012, 2013. And I was actually finding a lower concentration of this compound. Okay, so there's an inverse relationship with snow depth. So here's a 2013-14. Nonylphenol was higher, snow depth was lower. So it seems inverse, right? And I got to thinking about that. The amount of this compound that we're using from year to year really hasn't changed that much. But when you have more snow, the concentration of it is going to be lower. It's almost like a diluting effect. So on years where we have a lot of snow, the concentration's not so high that you might see it in that bioactive range. But in drought years where we have a little bit of snow, you're going to see concentrations that might be in that bioactive concentration. So it's just kind of an interesting link up that I made with some of the things that have been going on around here. So the higher concentrations then would be in the spring thaw. We get snow pulses where we get like a melting event, and we got a lot of the snow coming down at once. And of course, spring is a critical period for development in animals. And so that's when they might already be stressed out by drought conditions, and then they're being also exposed to this endocrine disruptor. So there's sort of a and there's our little friend the toad from last week. Of course, fish and amphibians are at higher risk because they're in the water so much of the time that they're going to almost have that direct contact more frequently. Okay, so my current research, just to sort of bring you up to speed on where I am now, we're looking at the release of 4-nonylphenol from sediments and streams and lake beds. I've done some really interesting work with that I hope to share with people soon. And so how long does it take to release back into the water once it's reached the sediment? So stand by, stay tuned for more information there. And I just wanted to talk quickly about where we are at with the regulation of 4-nonylphenol. I think it's interesting that the European Union banned it almost 15 years ago. European Union sort of works on a different model from what I can tell. They say, okay, if this stuff looks like it's going to be bad, we're going to take it off the market. We're going to test it and see if it is or not. We're a little bit more reactionary, whereas we wait till we see that it's really bad and then we take it off the market. So it's kind of like a different approach to regulation, I would say. But the EPA has done a few good things. I think they've asked for a voluntary phase out in industrial laundry detergents. It's been taken out of regular detergent, so the stuff that we might use in our washing machine, that it's been removed from that, but it's still used a lot in industrial laundry. So they've asked for a voluntary phase out. That was actually about 4 years ago they asked for that, so that was still Obama administration. And then it's also been added to the toxic release inventory. So if there's a spill, they have to document it. They have to say how much, when, where, and they have to record it. So they're responsible for it. They're not approving any new uses of it, which I think is fabulous. And it's been added to the toxic substances control list. So you have to say that this is bad stuff if you're going to be using it. You have to tell your employees, that kind of thing. The downside. In 2010, we imported 380 million pounds of it. We're still using it. We're still using a lot of it. So it's not phased out as much as I would like to think it should be. Also, recently, they've made a special exemption for pesticides. Yeah. So they're saying that if you're using it in pesticides, it's okay because you really, really need it for pesticides, apparently. So I don't feel like I've painted a particularly pretty picture. I feel like there's a lot of reasons that people can look at something like this and feel a little bit hopeless and a little bit down. But I also want to say that I feel like there is also a lot of hope. And that was Gretchen's message 2 weeks ago, which I really appreciated. Somebody who looks at ocean acidification can still say that she feels hopeful about things. So I'm going to say that I also feel hopeful. I feel like there's a lot of interest in this. I think there's going to be a lot of people that are studying it. And this is really what gives me hope. Yeah. Right here. These are the guys that are going to go out and change the world. So I just had to give a shout out to this year's lab group, my graduating seniors. Every single one of them worked with nonylphenol and ways to remediate it, get it out of different environments, and they've just done wonderful work. So, yeah. Anyway, with that, I just want to make a few thank yous before I take questions. First of all, the University of Redlands Chemistry Department, fantastic place to work, top of the line instrumentation. It's all research grade. The kids all get to use it when they're doing the research, so they all go out of there with some pretty serious skills. Summer Science Research Program, they get paid to do research over the summer, good salary, room and board, fabulous experience. I also want to give out a shout to Tony and Cheryl Taylor, who have supported my class and my research since its inception up here. They're kind of my Sierra parents. So I have to say thank you guys, for the geology tours and the dinners and everything. The Keck Foundation and the Center for Spatial Studies, they've supported me, the Keck Foundation financially, and the Center for Spatial Studies with their innovative GIS skills. Esri did the Spatial Communities of Practice grant, so they've supported my work a lot. SNARL for letting us be here and run around the place like lunatics. Thank you so much. Team Lyons, you guys know who you are, and of course my husband, who's my best field assistant. So with that, questions? Yeah, right there, friend.
I'm visualizing, we've had, trying to protect the frogs lately, worldwide decline. And I'm visualizing a map that shows the distribution of frogs in the problem. And there's no problem at all trying to protect the frogs east of Bakersfield, going down the Kern River Channel, because they're all gone. They had this big void, nothing there anymore. I was wondering if any correlation, I think there probably is, with what you're doing here. And the thing there, they said it's pesticides off the farm belt that have just annihilated them.
So he wanted to know if the pesticides and the Kern River frog decline are related, which yes, they are. I don't have data on the effects of nonylphenol directly on frog populations. It's something that I would dearly love to get. And I'm not a biologist, and I need to recruit a biologist to go out and look at frogs and look at abnormalities in their reproductive cycle and physiology and that kind of thing before I could say, oh yeah, there's a link, because
It's only a viral wasting degree, that's the problem, but there's apparently really some other problems.
yeah, I think they're under a lot of stressors. Yeah.
Chemical question, what's the aqueous solubility of nonylphenol?
It's on the order of 7 parts per million. So it is mildly water soluble. I mean, for a compound like that, to throw that into a chemical term, there's a phenolic group on it. So that's enough to give it water solubility, a little bit of polarity, and it will go into water. My students hate it because it doesn't want to go into water. But
Right, how about, lipids, fats, oils?
oh, in that, it'll, yeah, as much as you want to put in there. It's got a KOW, so KOW, folks, is a way of measuring how much it will dissolve in fat relative to water. And it's, the higher that number is, the more it will dissolve in fats. It has a KOW on the order of 10 to the fifth. So yeah, it absolutely will dissolve in fats. As much as you want to put it into fats, it'll go into it. Yeah. Question in the back?
You said that your students were working on ways to remediate nonylphenol in the environment. What have they come up with so far?
I think at this point, it's almost like we're trying to find links between exposure and humans. That was one of their projects. Like, are we seeing increased rates of different types of diseases to the human populations that are exposed? So we're looking in places like in the Central Valley, doing a lot of statistical work on that. And really, again, like with the ecotoxicology, it's really difficult to make a firm connection. So we really haven't come up with anything there. Looking at sediment release, talking about wastewater treatment plants and where there's a lot of it there, like trying to think of ways, are there ways that we can treat wastewater, treat the sediments, so this stuff is removed before it gets into the system. And then I'm waiting for the right student to come along that can work on surfactant replacement products. So, yeah, sir.
One of the slides said there was 380 million tons or something like that imported.
Mm-hmm.
Is it the source of what it's made of, or regulation that keeps it from being made here?
It's a regulation that keeps it from being made here. So, but it doesn't stop us from importing it. So that's the loophole.
What's it made of, and where is it made?
It's a carbon product. Mostly, I think their major importer is Japan right now. In the papers that I've seen, that's sort of the one that comes up the most frequently. And then in terms of how it's made, it's polymer chemistry. It's made with carbon compounds. Yeah, in the back there.
Do you take measurements throughout the whole year, and have you found that it's pretty consistent throughout the year how much the compound is spread or changed?
It's in different compartments of the environment at different times of the year. So that was actually kind of interesting to me as well, that like surface water, like late summer is the repository for it. And then you sort of see this tapering off, and then you get that snowpack effect. So by the end of this, like a lot of the measurements that we're taking were like March, April, and that's when we're seeing it sort of build up in the snowpack to the higher level. So it depends on like the type of measurements that you're taking, but there are definitely like cyclical trends to it for sure. Yeah, good question. Yeah.
Is it taken out of reclaimed water?
It's not. No, it's not. There's currently no treatment for it. Yeah, it's there.
And with the pesticides, is it an organic pesticide?
I don't know. That's an excellent question, and I'll look into that. Because I know there's like natural pesticides that people use.
Yes.
That maybe don't have, that would be lovely if they didn't. I don't think those are used as much as the other ones are. Obviously, the ratio of other pesticides to the organic ones are not great, but yeah. Dave?
It looks like the model predicted lots of it, deposition at Nevada. Did you get any transects out that way?
Not yet. That's on the list for sure. Yeah. You had a question?
That was my question exactly.
Oh, okay, cool. Yeah.
I'd be interested in hearing about transects along strike of the Sierra. There are big variations in the topographic shielding along strike. So I think the area north of here, for example, where
Mm-hmm.
rainfall or snow gets through, would have 50 concentration.
Yeah, actually, I'm really excited about doing that myself. We're planning on doing that this summer. And one of the gals in the picture there, she actually developed a sampler that separates out the aerosol from the dust. So that's actually going to save us a lot of time. So while she could just put a sampler out in the field, and it's like a little pump, sits there and pulls those things separately out of the air. So we plan on doing that up in those regions. So woohoo. Yeah, thank you. Yeah.
The majority of snowpack sublimates
goes to gas.
Mm-hmm.
Does this material go back into the air, or is it supposed to be grounded until it rebounds to dust or some other condition elevates it, then sublimation?
Okay, so I'm thinking about its Henry's law constant, whether it has the capacity to do that. And I don't think it will. It's not a high enough volatilization rate for that. So I'm thinking, and actually I talked to somebody from the ASO group last year who was looking at how the snowpack melts, and really the dust from the snowpack ends up just on the ground. And so that means that this stuff will also end up on the ground. So when I was initially concerned with like, oh, there's going to be lots of it in the spring runoff, maybe not as much there as there will be like on the ground, and maybe the first few rain events would pull that out.
What separates it from dust?
So that's what we're looking at right now in the river and the lake sediment, is like that sort of over time. It does have some degree of solubility, like we mentioned earlier. So it will eventually reach sort of an equilibrium with the water that it's in. And it's interesting because it depends on the amount of carbon in the sediment or the dust. And so the higher the carbon content, the more it's going to stick to the dust. So it really varies. There's some variability there. It's pretty interesting, yeah.
Does it seem to bond equally well to water in a liquid state as it does to
The snow?
solid state? Yeah.
Well, I don't think so. And I think that's kind of part of the reason the snow is made such a good reservoir for it, because there's that surface area effect where it's picking up the aerosol particles as it's rhyming. And I think that's part of the reason that it's accumulating to the degree that it is. Yeah.
Is snow pretty efficient at scrubbing?
It's excellent at scrubbing the air. Yeah, those papers, I think it's Wania and Mackey. It's a really good paper to read about that, because it's pulling all kinds of stuff out of the air. It's fascinating. So, yeah.
Has it been found in drinking water?
Well, it's been found in reservoirs that supply drinking waters. I haven't tested, I didn't even think to test it out of a tap.
And so if it would carbon filter?
Like a carbon filter would do a good job, like a Brita filter, right? Because it's going to selectively bond to that.
Or carbon filtration in a water treatment plant?
Potentially. Yeah, potentially. Although, I have tested wastewater effluvia in a couple of different places, and it's always present in just huge amounts in wastewater effluvia. So the stuff coming out of a wastewater treatment plant. Yeah, it's almost always there. Yeah.
Do you have plans to look at bioaccumulation of
The 4-nonylphenol?
in the variety of organisms in lakes or stuff that you can correlate to the distribution away from the Sierra Nevada rainforest?
Such a great question. Yeah, I'm so not a biologist, and I'm so skeeved out by those kinds of studies that I know I need to recruit a really good biologist who wants to work on this stuff with me. But I know there was people who were looking at like breaking down the different types of tissues and looking at it in organisms that like maybe have a higher percent lipid content and that kind of thing. That would be really interesting, though. Yeah.
Throughout the traveling of this chemical, does it retain its structure? And does it also tend, because it's lipophilic, does it also tend to congregate on surfaces? Or is it more, does it adhere more to like the carbon?
Yes, it's an awesome question. That's actually, I think, going to be in the next study that we're doing right now. So the congregating thing, I'll answer that question first. We found that when particles clump together, they actually, so like a lot of the times if you've got stuff in a stream, it's not like you're just getting individual little particles. They'll sort of ball up together. And so what that does, it actually creates little pockets of space where this stuff can kind of hang out. And so we've seen that in activated carbon when we've done that in the laboratory, and we've seen that in just dust when we've done that in the laboratory as well. So as soon as there's some sort of agitation, you almost create these little places for it to hide, which is interesting. As far as the structure, it does retain that structure. I said it was the most energetically favorable of the potential arrangements that you could put those carbons in. So in terms of like what it's going to go to naturally, it will always go to that structure. That's going to be the easiest one for it to form. Yeah.
You said early on that the estrogen receptor was not particular.
Mm-hmm.
In nature, is it so unique that there is little else that would want to fit into that receptor?
There's a lot of estrogen mimics. And I think that to answer that question, I think there are a lot of other things that will fit into that. So the chart that I gave you guys, I think 4 out of the 6 of them were estrogen mimics. So it's just like I said, it's not so selective that things can't bind to it. It doesn't even have to bind that strongly for it to have an effect, for it to have a biological effect. Yeah.
Well, isn't it kind of hydrophobic?
It is hydrophobic. Exactly what it is. So it's gonna love skin, fatty tissues, anything like that. Yeah. Yeah.
So realizing the problem is much bigger than the kind of products that we might buy for our own home.
Mm-hmm.
But as consumers, does it kind of make sense to look at the list of things and what would we look for again? Not so ideal.
That's awesome. I really wish I had a great answer for that one, but I think a lot of the times, I know that they've been phased out of detergents projects that we would buy for ourselves. We still find them in sunscreens, and so a lot of the times they aren't required, or like lotions and stuff. They're not required to say what that stuff is in there. They'll just say inert ingredients on there. I tend to lean towards things that are just organic, where they list absolutely everything, and I don't see that word in there. So if they're covering their tracks by saying inert ingredients, I don't buy it. So there you go. Yeah.
There's a website, I'm not remembering what it's called, where you can look up.
It's called Green Screen. Yeah, and it's a really good website. Yeah, thank you for reminding me of that. Yeah. Greenscreen.org, I believe. Yeah.
What was the website again for the
Oh, the wind map?
Oh, yeah.
Oh, yeah. So cool. It's the fun part.
And maybe we'll take one more question, because I know we're wearing Rebecca out. This is we can ask questions all day on this stuff. It's fascinating.
Yeah, it really is.
If there's one more, and then maybe what we could do is let Rebecca go. Is there one more pressing question? No? Okay.
Guys, thank you for your interest in
Yeah,
this.
thank you, Rebecca.
It's awesome.